Hello everyone, and welcome to our third webinar, part of our webinar series on health application from marine ingredient. I'm really pleased today to welcome our three amazing speakers who will introduce their research about stress management. And with all this COVID situation, we can say that it's really a hot topic. And now people are looking more and more for new natural ingredients to support their health. My name is Rachel Durant, and I am the project manager of Marine for Health for the cluster NCE Blue Legacy. Blue Legacy is a Norwegian cluster, and we are located on the west coast of Norway at Ålesund. The purpose of the cluster is really to improve and increase the use of all marine resources to produce new high-value products for human consumption. Now we have more than 50 members from all the value chain, so from the fishermen who produce these new resources, from the fish plants, and also from the final ingredient producers. The Marine for Health project aims is to establish new scientific expertise for the health effect of these ingredients, but also to documentate these new resources. We also support our members for the regulation part and also in their R&D journey. Before to start the webinar, I just remind you that it is recorded and the video will be available on our website. So Julie Lefaudet is the peptidomic project manager at Abyss Ingredient, and she has over 15 years of experience in the analysis of biomolecules, including 12 years in proteomic in the health sector. She is part of the R&D team at Abyss Ingredient, where she is in charge to characterize bioactive peptides for the health nutrition. We also have Claire Legrand. She is the sales manager at Abyss Ingredient, and she started her career in the food industry in the east part of France, but she moved back in the west part at Brittany to work with the idea to prevent waste production by creating new natural ingredients for human health. So now, Julie and Claire, the floor is yours.
Thank you very much, Rachel. Thank you for your presentation, Anita. It was very interesting also. Can you see my screen right now?
Yeah, perfectly.
Perfect. So thank you very much for your invite to this webinar and letting us the opportunity to present Abyss and what we do in stress management ingredients. So basically, Abyss is a French biotech company. So we are based in Brittany, just like Rachel said, and our mission and our aim is to evaluate local fish byproducts in order to develop innovative, natural, eco-responsible and sustainable ingredients dedicated to health, human health, and more specifically the dietary supplement market. And of course, Julie and our R&D team are dedicated to the scientific proof of efficacy of our ingredients. So our ingredients respond to specific needs of the aging population. So mobility with joint and bones care, beauty, but the ingestible beauty thanks to the food supplements. And since a few years now, we invest a lot in the cognition application, as it is a growing demand, and we are aging. We are not getting younger, so we invest a lot in cognition and especially stress management, sleeping disorders, and memory troubles. Um, Rachel, you said it. Stress management is a growing concern, especially in the actual and the current context of sanitary crisis. So just to give you some figures, in before the COVID crisis in 2019, the stress and sleep management dietary supplement market was already increasing by roughly 5% in France. So it is a France figure, but it is a worldwide concern. And there was a huge boom at the first semester of 2020 in this application due to the COVID crisis, of course, as almost 60% of the global population said they were stressed due to the coronavirus situation. But there were not only this boom of stress and sleep application demand. There is also an increasing demand for natural health products, and globally there is an increase of food supplements compared to the previous years. And people are searching for more and more natural, sustainable, eco-friendly products. So I will get back to our subject today. So what is stress is exactly? So I think Anita Lill-Hansen knows it better than me, but basically it is first of all a natural phenomenon, and it is important not to avoid this response. We have to maintain the stress response because it is a natural physiological reaction from our body when we are facing a situation of a stressful situation and pressure. So basically, when there is a stress, there will be a sequence of response from our hypothalamus to pituitary gland to adrenal cortex, and then there will be the release of the cortisol. Of the cortisol, which is the stress hormone, that will lead to negative feedback, and then return to a normal level of stress hormone. But of course, as everything in this world, there is good stress and bad stress. So the good stress, the positive stress is when the stress is punctual, and it brings us some good energy and adrenaline. But when it's not punctual anymore, when the situations of stress are repetitive, then it can lead to some disagreement, like sleeping disorders, bad mood, etc. And if the situation keep on going with bad stress, then it can even lead to acute chronic disorders such as weight management, hypertension, depression, so more severe troubles. So of course, there are some solutions to avoid these severe disorders. The first one is the cognitive behavioral therapy. So basically, you can use some relaxation, yoga session, etc. to deal with your stress. So it works when you're not too much stressed. Of course, so France and French people are the champions in this field, unfortunately. Um, you can use some pharmacological treatments like anxiolytics, but there are some risk with using this, like dependency, and yeah, there are drugs actually. So there is an innovative approach, another strategy that you can use. It's nutrition, and dietary supplements are part of nutrition. So many publication and research already demonstrated the effects of plants on stress and sleeping disorders, for example. But there are also food hydrolysates, which presents anxiolytic-like peptides, which present the advantage of having fewer adverse effects, lower risk of dependency, and there is a good bioavailability. And of course, the main part of this food hydrolysates are totally natural. So here we are with our peptidis, which is a natural, just like all our ingredients, so natural and sustainable ingredients. So it is produced from local sardine byproducts, so wild catched in Atlantic Northeast waters, and then we process it through enzymatic hydrolysis, which give to our peptidis a unique composition, mainly composed of proteins, some minerals, but here we will talk about peptides, which have a low molecular weight, and it makes our peptides highly digestible and bioavailable. So as I said previously, since few years, we invest a lot in research and development in order to demonstrate the efficacy of our ingredients. And we led in 2002 a first preclinical study on peptidis to demonstrate its anxiolytic-like effect. So we had 5 groups. So 1 control, 3 dosages of peptidis, and 1 positive control with the diazepam, which is an anxiolytic drug, which is very well used and known in France. So basically, we supplemented so with high dosage of peptides, but still we supplemented the animals 1 hour before the test. Then we get them through a high a noxious stimulus, which is intense stress, and we determined the global anxiety score of our rodents. And here we had, so here is the effects that we evaluated. So basically, you can see that here there is a significant effects of peptidis on the stress reaction. And we can even say that peptidis has a diazepam-like effects. So diazepam being a medicine, this is very encouraging. So to confirm this test, we led a second preclinical study 2 years ago in 2019, and Julie, my colleague, will present it to you.
Thanks, Claire. So the objective of this second preclinical study was to evaluate the effect of a peptide supplementation on a daily life stress. So the study was performed on 4 groups: peptidis stressed and peptidis non-stressed, control stressed and control non-stressed, with 11 individual per group. After 1 week of supplementation, mice were submitted to an acute mild stress protocol. Blood was collected before the beginning of the stress protocol and after 30 and 60 minutes of stress. Then the level of stress hormone, corticosterone in mice, was measured in plasma. So our results demonstrated that 30 minutes after stress induction, peptidis supplementation decreased the level of corticosterone compared to the control group without blocking the stress response that is necessary to cope with stress and adversity. The first evidence demonstrates that peptidis can reduce the deleterious effect of stress. In the second part of the study, we evaluated the impact of peptidis supplementation on some of stress-responsive genes expression in specific cerebral structures, hippocampus, prefrontal cortex, and amygdala, which are involved in the regulation of stress, cognition, and mood disorders. The principal component analysis of these genes evaluated in hippocampus demonstrated that the expression of gene in stressed animals supplemented with fish hydrolyzate was different from that of the other groups. We demonstrated that peptidis supplementation modulated genes associated to stress pathway like SGK1. In a matter of fact, the upregulation of these specific genes can act as a primer of corticosteroid signaling. Moreover, peptidis supplementation also regulated one of the core genes of the circadian clock, PER2, playing a critical role in sleep, especially in familial advanced sleep phase syndrome. As stress and sleep are connected and codependent, the upregulation of the gene by peptidis supplementation could participate to its anxiolytic effect. Taken together, those results demonstrated that peptidis can be an innovative strategy to regulate and manage stress. We also demonstrated that peptidis modulated SIRT2. SIRT2 plays diverse roles in cellular metabolism and aging, and the connection between SIRT2 and neurological disorders is now well established. The potential of SIRT2 as therapeutic target in neurodegenerative disease started to be studied. The upregulation of SIRT2 by peptidis in basal and stress conditions could be of great interest to prevent neurodegenerative alteration and should be further investigated. The whole study has been published in 2021 in Journal of Functional Foods. From the same cohort, we are now investigating the proteomic profile of mouse hippocampus to comprehensively understand the proteomic changes induced by peptidis supplementation in acute mid-stress conditions. For that purpose, we performed a label-free quantitative proteomics analysis, which consists in digesting protein with protease in order to generate peptides. Peptides are analyzed by nanochromatography coupled to mass spectrometry. Generated data are confronted to protein database for the protein identification. Then the relative quantification of proteins is performed according to the 4 groups: control non-stress and stress, and peptidis non-stress and stress. We are currently processing the data. Abyss is also implicated as project holder since 2018 in a 3-year collaborative project, the VIP project for very important premium peptides. The objective of this project is to identify the bioactive peptides responsible for stress management in peptidis. For a better knowledge of our peptidis solution, and in order to launch a new premium ingredient with an enhanced efficiency, and finally to develop new nails application. This collaborative research project is backed and funded by the European Regional Development Fund and Région Bretagne organization for a total investment of 1 million euros. This project is led by academic partner, pro team from the Rennes University, and industrial partners, Abyss Ingredient, Capsularis, Ceres Pharma, and Idemer. Each expert partner contributes to the project as follows. ProTeam and Abyss Ingredient are developing peptidomic methods based on mass spectrometry and bioinformatics for the identification of the bioactive peptides. MS analyses are performed on the latest generation device comprising the ion mobility separation. Ceres Pharma is elaborating in vitro efficacy tests for the assessment of anti-stress activity. Capsularis work up microencapsulation techniques to improve the organoleptic properties and the bioavailability of the peptidis hydrolysate. Idemer will optimize the manufacturing process to generate the premium ingredient with enhanced efficiency. And finally, Abyss will launch a new premium peptidis. And the exploration of these different areas should open up new applications.
So we keep investing in science, as you can understand, and we will continue by the Graal in dietary supplement market, which is the clinical studies. So we just launched a clinical study which aims at evaluating the stress and sleeping disorders management effects with peptidis. So this study will be launched in partnership with DACRO, which is a CRO specialized in stress and sleeping disorders. So this clinical study will aim at confirming the preclinical studies and results that we had on both stress management and sleeping disorders. So it is a randomized double-blind crossover vs placebo study. It will last 4.5 months. It will be led on 44 people, men and women, perfect 50-50, and age between 35 and 60 years old. And evaluated dosage will be the dosage evaluated, the equivalent dosage in human evaluated on our preclinical studies. So we are impatient to have these results that we plan to have by the end of 2021, so this year. Thank you very much for your attention.
Thank you, Claire and Julie, for this amazing presentation. And I'm really looking forward for your last study, and I hope you will be able to present us the results.
With pleasure.
Yeah. So now we will have a short Q&A discussion. So please, you can use the chat or the Q&A to ask some questions to our speakers. Maybe Claire and Julie, you have some question for Anita? If not, I have some.
Yes, perhaps just one question. Anita, are you going to investigate the mechanism implicated in the effect of the vitamin D supplementation on stress? The mechanism, the biological mechanism implicated in the effect?
Ska man säga, what did you say? If I'm going to investigate the effects of vitamin...
to know how vitamin D acts playing a role in the regulation of stress?
Yes, well, that was what we tried to do with the resources we had. But as you might saw from the presentation, vitamin D did not affect cortisol in this sample, but it's a very special sample. Actually, these participants had from the beginning very low cortisol responses compared to normal healthy subjects, because they have so many problems, and many of them have been exposed to trauma. So it could have been interesting to look at use the same design on healthy participants.
Of course.
Yeah, actually, it was my question. Why choose this? Because it's really a specific population, and I can imagine that they have a basal stress. So why choose this population? What did you start by them?
That was because that I'm from the beginning we're working with people with behavioral problems. So and the effect of psychological treatment in these patients is not so good. So that's why I wanted to see whether nutrients and nutrition could boost the effects of traditional psychological treatment. Because if they have been exposed to trauma, these stress response systems are destroyed. So the main idea is to see whether nutrients can, in a way, help and calm down the physiological activation.
And I have also a question, because of course the winter season we know in Norway and in the Nordic country is really for me it was really the first time it was really a stress situation, and it's really different from the other country. But do you think that you can have the same result or the same benefits from fish consumption for also France or more south countries?
Yeah, the thing with fish is that there's so many important nutrients in the fish. So maybe it's just not the vitamin D. Actually, I think that it's the fish as whole food, because if you're eating fish as for dinner, you will replace fish with something unhealthy, for instance. So actually, I have stronger beliefs in fish than supplements. But we thought that if we should find more out about the effects of vitamin D, we have to do a robust vitamin D trial to find out the effects of only vitamin D. But I believe in fish as whole food. Yes.
But actually, it's always the same challenge. You can have a good first health effect on a food product, but it's difficult to find really the specific compound and the bioactive compound. And actually, I have a question for that, for Abyss ingredient, because now on the fish hydrolysate or protein, any protein hydrolysate, there is a debate about the synergy between peptides. My question is, it is really useful to use only one peptide, or is more useful to have a composition of different peptides?
I can try to answer. It's a very good question. I think we try to find some bioactive peptides by fractionate our hydrolysate and to show the activity of specific fraction in order to concentrate our product with a bioactive peptide. But it is a very complex mixture with thousands and thousands of peptides, and probably there is a synergy with all these peptides. So we are still investigating this point, and we can see that there is an effect. We decrease corticosterone level, so there is clearly an effect. And is it one or several peptides? The synergy, we are still investigating this point.
Yeah, but it's really an important also a step, this concentration, this peptide concentration. We have a question from the chat. Maybe it's for you, Anita. What is your recommendation or advisory on a fish-based diet for people between 20 to 30 years old?
Yes, I'm not a nutritionist, so I think that I will advise you to look into the website of the health authority suggestions. Usually it has been about 2 fish meals per week, but I think you should go into the health authorities advice. Yes.
Yeah, I know that for the omega-3 is between 250 and 500 milligram, but for the vitamin D, I'm not sure also.
Well, I think it's about 38 microgram is the recommendation. Yeah.
And so you can find this dose in an whole fish, in one whole fish, or you need, I'm suppose you need to eat more than one?
Well, I think you have to eat a lot of fish to get the right amount of vitamin D. But the thing is that, as I said, fish is a rich source of many other nutrients other than vitamin D. But I think that you can eat very much fish. So I think during the winter in Norway, you should take some supplements in addition to,
the cod liver oil.
Yeah.
So we have a question also, and actually it was also part of my question. My question was, what did you choose sardine? And the question from the attendees, what part of the fish did you choose? Did you utilize? Sorry.
So why sardine? It's actually we came from we developed peptidis from the scientific literature that we have, and we know that some peptides are involved in stress management, and so we demonstrated it. Which is a first step. Then the second step, as Julie said, is to identify and characterize the peptides that are responsible for the stress response and the stress activity. And regarding the part of fish, as previously said, we use the local fishing industry byproducts, and so as you may know, in Europe we have some regulation regarding fishing, and sometimes there are very small sardines that are catched and that can't be used for human consumption as food. So we use these small sardines that are too small to be used as food. And we also use some sardines plants. So they take the fillets, the fishing industry take the fillets for human consumption, and then it remains the plants, and we use also these byproducts.
So you use all the fish, but also really specific part.
Yes, exactly.
But for the same product.
Yes, so we have a product specifications that we have to respect, of course, and we always make sure that we are in the lines what we do, and but yes. Yes, our first mission was really to evaluate byproducts, marine byproducts, and to decrease the volume of waste that could be induced by the fishing industry. And yes, this is what we do.
Okay, thanks. It's also an important issue in Norway, and we try to use all the fish, not only for aqua food, but also for human consumption. And I think it's really important to improve this use. I have a specific question for Abyss ingredient. In your presentation, we saw one result where I think it was your first study, where a high dose of peptidis was less effective than low dose. How can you explain this kind of results? Because I know that in some paper also you can find sometimes these results.
Maybe I'm not. Maybe Julie is better has a better knowledge to answer. But what I understand from this result is that so first of all, the three dosages are very high dosages, and what we can see is that it is not necessary to have a very high dosage to have it doesn't increase or improve the effects. It's just like if we had I don't know how to say, like a level like the perfect dosage, and once you go above, then there is less effects. So yeah. What is interesting, because in the food supplement market there is there was a question of what is the perfect intake or the ideal intake of fish per week, per day, etc., to have effects on our cognitive health. In a dietary supplement market, it is important to know that the lower dosage, the better it is, because our clients who are laboratories and contract manufacturers, they have to fulfill small capsules or to make some tablets, etc. And it is I don't know if you use some food supplements in your daily life, but it is not very easy to have to take like 4 or 6 capsules per day. And here the idea is to have the lower dosage possible. And so what is interesting here in this results, it's that with a moderated dosage, you have effects. And this is what we want to show.
But yeah, actually this Bell effect, I don't know if we can call that like that, but yeah, sometimes in some dosage test, we can see that high dose can be toxic. And actually sometimes small dose are more effective than a really high dose. Okay, thank you, Claire.
May I just add regarding the toxicity? So there is so we had some toxicity tests on peptides, but you would need to eat a lot of it to be sick or to get healed. So once again, it's a natural product, so there is no toxicity issues.
Yeah, I can imagine. I think I have a last question, and I think it's for everyone. With all this presentation, we saw that to evaluate the stress can be really difficult. There is a lot of different mechanisms, different situation. What for you as a researcher is maybe the most important thing to evaluate? with the stress? Maybe Anita, you can start.
What do you mean? What is the most important to evaluate in relation to stress?
Yeah, because we saw that you can evaluate the rate of your heart, you can evaluate
Yeah.
some hormone, you can evaluate also some connective test, some gene also we saw from Abyss ingredient. So for you, what maybe it's the first thing to do?
Well, actually, I also think that it is important to evaluate the subjective experience of stress, because as Claire said, some kinds of stress can be regarded or experienced as positive, other kinds of stress can be regarded as very negative. So talking about stress and evaluating stress, I think it's also important to ask the subjects, did you feel this stressful or not? In my experiment, I used an experimental stress procedure. And based on the psychophysiological responses, it was stressful. But we don't know whether they experienced it as stressful. So, yeah. So I think it's important to get the grasp on both the subjective experience and the physiological experience of the stress.
Thank you. And for you, Julie and Claire, what you think is maybe the most important?
For Abyss ingredient, it's a complementary approach. We made a preclinical study, and now we are going to start a clinical study that I think it is the best way to prove the effect of our product. But with preclinical study, we can have more information on genes and proteins changes. So we prefer explore all the way, and with a clinical study, we will be more focused on the final results. But
yeah, and we saw that you start your the first study, it was 2002, so it takes 20 years to develop a new product. So it takes time.
Yes, because there is a big change in Abyss ingredient, and we are 3 or 4 people now who arrived in Abyss in the research and development team. So now there are a lot of projects which begin last year.
Great.
If I may add, I think that the stress reactions in our body is so there are so many ways to evaluate it, because our body is so complex and there are so many connections between all our hormones and cells and behavior that I think it's there is a full picture, but we cannot. It is so large that we cannot have the full picture at once. So we have to explore several ways, and this is what. So we had some ways within Abyss, and Anita is I think more exploring. Also the psychological points, and all is complementary, and this is very interesting.
Yeah, and that's why also as a cluster we know that to develop this kind of project, you need a good partnership and a good collaboration, and we try to do that. So I think in this with this last word we can finish our webinar. I want to thank our speakers for this their amazing presentation. And now we know a little bit more on stress, but I think we just scratch the surface. As I told you, each presentation will be available on our website, and I'll also invite you maybe to subscribe to our newsletter to be aware of our next webinar for health application for marine ingredient. So thank you everyone to attend to our webinar, and I wish you a great day. Bye-bye.
Thank you.